Bronwyn, look at the. Oh, okay. It's already. It's been resolved. Everything's all right. So, welcome back, everybody who has chosen to join us today to the second installment of of this year's Actino-based e-seminar. I am proudly presenting today Bronwyn Kirby-McCullough from the Institute of Microbial Biotechnology and Metagenomics. To shortly introduce her, she did her PhD in microbiology at the University of Cape Town in South Africa. And after that, she moved to the University of the Western Cape, where she first did her postdoc, then acquired a position as an NGS manager, and in the end has now the position of a lectureship. And she will talk to us about the screening of antibacteria associated with indigenous South African plants for natural products. So Bronwyn, as soon as you're ready, the stage is yours.
Good afternoon, and thank you for that introduction. So welcome from a very wet and cold Cape Town. Like I was introduced, I'm Dr. Bronwyn Kirby-McCullough, and I'm a PI at the University of the Western Cape in Cape Town, South Africa. And today I have the really great privilege of being able to present on behalf of my student, Mrs. Maria Katrin King, who unfortunately is not able to present today due to work commitments. She's one of the wise ones who's escaped to industry. Katrin was actually my group's first PhD student, and I'm very happy to say that the examination process of her thesis is very near to completion. So we will know the results in the next few days. And Katrin did a really great job in the lab, and so I'm really happy to be able to share her work with you today. So like I said, the title of the talk is Screening Actinobacteria Associated with Indigenous South African Plants for natural products. Okay, so first formed within the Institute for Microbial Biotechnology and Metagenomics. IMBM is one of the leading research units
I'm sorry, Bronwyn. In case you can hear me, we seem to have a little technical issue with connection to you. Maybe we should just... Okay, we can see you again.
Okay, cool. Sorry,
I'm really sorry. I didn't want to interrupt you there.
No.
So
cool.
I guess we just try going on, and I will try to just inform you in case this happens again. I was just not sure if it was on my end, but apparently
okay, cool.
It's not. So please continue.
Yep. And so we about 35 researchers, including.
Sorry to interrupt. We've lost your slideshow.
Oh, okay. Sorry. Let me. Sorry.
Perfect. Thank you.
So we're about, like I said, about 35 researchers. We have 4 PIs as well as our own in-house research and innovation manager. So actinobacteria are naturally found in soil and can be easily interact with the plant roots within the rhizosphere. Here they form these symbiotic associations with the plant. From the rhizosphere, they can actually also enter directly into the roots, and then they become endophytic. And once they're endophytic, it means that they can actually colonize the internal plant tissues. When they've entered the roots, they can also then translocate to other parts of the plant, such as the leaves. In addition to these endophytic organisms, we also have what we call epiphytes, and those are organisms that don't penetrate the tissues, but rather just live on the surface. And this area that they colonize is called the phyllosphere. So these are the upper parts of the plant. Studies have shown that there is the greatest diversity of endophytic actinobacteria associated with the roots. And then the number of organisms decreases as you move from the stems, with the lowest diversity you find in the leaves. So the really interesting parts of it's always what's going on in the rhizosphere, and mainly because these rhizospheric organisms form really complex mutualistic relationships with the host plant, and they've actually been found to be essential for plant health. So there's several ways in which microbes can improve the host plant growth. They can do what we call either direct mechanisms or indirect mechanisms. Your direct mechanisms are ways where they directly improve growth, normally by aiding in nutrient acquisition, producing siderophores, fixing nitrogen, or producing plant hormones. On the other hand, the indirect methods of improving growth is by doing things like producing antibiotics or antifungals or cell wall degrading enzymes. And these can obviously just protect the plant from predation and pathogens. And you can clearly see that given this wide array of metabolites that these organisms are producing, plant-associated microbes are a really attractive target for any microbiologist or biotechnologist who's looking for novel natural products. So given that there is an estimated 22,000 plant species in South Africa alone, how did we as a research group decide where to start looking for novel bioactive microbes? So in their landmark paper in 2003, Strobel and Daisy listed 4 criteria which they said you should consider when selecting plant species to screen. The criteria were looking for plants that are from a unique environment, screening plants which are endemic, or plants that come from an area known for its biodiversity, or lastly, focusing on plants with an ethnobotanical history, where ethnobotany is defined as the use of plants in religion, folk medicine, or traditional healing. And for us, living in the Western Cape, an obvious region which fulfills all of those criteria is the fynbos biome. So South Africa, though it's a small country, has a very diverse climate, and this has divided the country up into 9 major terrestrial biomes. The smallest of these biomes is the fynbos biome, which encompasses the Cape Floristic region. This CFR is the smallest of actually the 6 global floristic kingdoms, though despite its small size, it is also actually the most diverse of the kingdoms, and it has the most diversity per unit area. The plant diversity within the fynbos biome is said to rival that of the Brazilian rainforest. There's also much less chance of finding really scary anacondas in South Africa. So anyone who's interested in plant diversity should definitely consider coming to South Africa for a visit. So for those of you who may know a little bit about South African geography, the fynbos biome extends from just past Klein Willem along our west coast, all the way along the east coast to just past Port Elizabeth. And the entire surface area of the biome is about 90,000 square kilometers. So despite its small size, there are about 9,000 species which are found within this biome, and 80% of these are endemic. So the characteristic plants that you find within the fynbos biome are generally small, hard-leaved evergreen shrubs, and these normally depend on fire for their propagation. So the popular floral species people would know when they think about fynbos are probably your proteas. So these are your beautiful pincushions as an example of a protea, your erica, which are these very dainty little flowers, and also things like veitjies. About 3,000 South African plant species are used medicinally. And they estimate that about 72% of South Africans make use of some form of traditional medicine. To give you some idea of the scale of the traditional medicinal plant trade in South Africa, it's estimated to be about 2.9 billion South African rand, and that currently equates to 146 million pounds per year. So as part of Katrine's study, she focused on 2 plants. So the one plant is Southerlandia frutescens. So Southerlandia is an upright Southern African leguminous shrub. It's most commonly known as cancer bush or kankerbossie in Afrikaans. This is a perennial, short-lived shrub, and it forms these very distinctive purple, bright red flowers. And then the fruit sacs form these unusual, large, bladder-like papery pods. Its distribution is restricted to the drier areas of Southern Africa, where it grows in savanna and hillsides near streams. The common name cancer bush is derived from the fact that historically, both Khoisan communities and Dutch people used it to treat internal cancers. So cancer bush extracts have been used to treat a wide range of ailments, from stomach ulcers, inflammation, diabetes, rheumatism to chickenpox. Traditionally, it was consumed either as a tea or a tincture, but now you can actually find it as supplements and tablets. The key metabolites that you find within these extracts are the canavine, the GABA, and 2 unique compounds are the terpenoids, Southerlandolins, these guys over here, and the flavonoids, which are the Southerlandiosides. Studies of Southerlandia extracts have demonstrated that it has anti-prolific effects on human breast cancer and leukemia cell lines. They've also been shown to induce apoptosis in Chinese hamster cells, as well as esophageal cancer cell lines. And extracts from the flowers and the leaves has also been shown to inhibit HIV reverse transcriptase, as well as the glycohydrolase enzymes. And this has subsequently been shown to actually reduce the infectivity of the HIV virions. The other plant we focused on in the study was Aloe Ferox. So Aloe Ferox is also known as bitter or Cape aloe. This plant is indigenous to the Cape coastal regions. Aloe Ferox is a single-stemmed, hardier plant that grows to about 10 meters tall, and it can be characterized by these very distinctive, very fleshy leaves, which have these very red-brown thorns. And that's actually the reason for its botanical name, Ferox. Ferox meaning fierce or ferocious in Latin. During the winter months, so at the moment, actually all campus is surrounded by these beautiful plants in bloom. The Aloe Ferox produce the single-stemmed racemes with bright red pink flowers. My apologies. Which are very distinctive. Aloe Ferox is also one of the few plants which were actually depicted in traditional sand rock paintings. Traditionally, Aloe Ferox has been used as a laxative, as also it's been used to treat skin ailments, as well as for the treatment of cancers. Leaf extracts have potent antioxidant properties, and Aloe Ferox has actually been incorporated into numerous cosmetics, medicine, as well as health food products. So the leaf of aloe can be divided into 2 parts. You have your outer rind, and then your inner pulpy, fleshy bit. The bitter aloes, which is the part that we use a lot medicinally, is derived from this pulp, which is then dried down into a really hard black resin. This resin contains aloe resin A, aloe resin, and aleon. The orthonon C glycosides, aleon A and aleon B, are found in the bitter part and is responsible for the bitter effect as well as the laxative properties of this product. This clear pulpy gel is also used for its emulgent and moisturizing properties. And research has actually been able to prove that this pulp can prevent some skin redness, it has anti-inflammatory properties, and it can also inhibit the growth of C. acnes. The other compounds you find in the pulp is your aloemoidin and aleon A, which has been shown to prevent the growth of a wide range of pathogenic gram-positive and gram-negative organisms. It can also prevent the growth of fungal spores of the Cryptococcus neoformans. So brings me to the aim of Katrin's study. So this is my very happy PhD student in the natural environment. So Aloe Ferox and S. frutescens are indigenous to the South African fynbos biome. And given this high level of plant species diversity, as well as endemism in this region, it is hypothesized that the actinobacteria associated with these medicinal plants have the potential to produce unique compounds of biotechnological interest. And this research aimed to isolate novel actinobacteria associated with these plants and screen them for natural products by coupling your traditional culture-based methods with genomics and bioinformatics to help identify the potential compounds. Okay, so just a brief overview of the methods. So the project was divided up into 3 main parts. You have the isolation and identification, then the antibacterial screening, and then lastly the genome analysis. So for the isolation, we got Aloe Ferox from organic aloes in Albertina, and Southerlandia plants we got from farms in Wellington. The plants, tissues, roots, leaves, and seeds were all surface sterilized and treated, and then we plated it on a range of media, including humic acid vitamin media, starch casein nitrate, yemi supplemented with leaf extract and soil extract media, and as well as actinomycete isolation media. Isolation media is also supplemented with antifungal agents. The plates were incubated at room temperature for about 5 months, and identification was done using your standard 16S and GRSB sequencing, as well as some polyphasic testing. For the antibacterial screening, we started by doing a perpendicular streak test. Their actinos were grown for 10 days on yemi, and then we tested against Bacillus cereus, E. faecalis, E. coli, and Staph aureus. Overlays, so perpendicular streaks was followed by overlays, where we did testing of 14-day-old cultures on three different media. We then did solvent extractions, where the actino was cultured in either ISP1 or ISP2, and metabolites were extracted from the cells using methanol, and from the culture filtrates using ethyl acetate and chloroform. We then tested the activity of these extracts using the Kirby-Bauer method, and we tested it against an extended range of test organisms, and those which were shown to have activity were then analyzed further using bioautography. We also screened all the isolates for siderophore activity. The last component of the methods was looking at the genomes. So we first used the PCR-based screening method, where we used KS1 primers to look for the presence of PKS type 1 genes. We then did whole genome sequencing on an Illumina MiSeq. The genomes were assembled using CLC Genomics. These were annotated using RAST. Biosynthetic gene clusters were identified using Antismash, and we also determined what the ANI was. Okay, so for some of the results. Katrin was able to isolate 241 isolates from the Aloe Ferox rhizophyric soil, as well as 5 endophytic strains. Not surprisingly, most of the highly active strains were streptomycete species, but we were able to isolate an Amycolotopsis, as well as an Aloe-actinoskinema species, which was really interesting because currently there's only 2 validly published species within this genus. So that was a really interesting find. Based on the 16S RNA sequence similarity, it does appear that there are several potentially novel species, which Katrin didn't have time to analyze. So these are currently forming the basis of an honors project going on in my lab. When we looked at then what we found growing in the Southerlandia, Katrin was able to isolate 172 actinos from the roots, as well as 6 leaf epiphytes, 4 leaf endophytes, and 6 root endophytes. When you look at just in the top hit list here, you can see that the species diversity was much wider in the Southerlandia plants compared to the aloes. And we were able to isolate streptomycetes, Actinomadura, Amycolatopsis, as well as Micromonospora species. As with the aloe species, it appears that several of these species are novel, and work is ongoing on these strains. So as we had such a large number of isolates to screen, it was decided to use a traditional perpendicular streak method as our primary screening method. So to do this, isolates were grown on Yemi for about 10 days. And after that, they were tested against the Bacillus, the Staph, the E. coli, and the E. faecalis. So this is just the control plate showing that the test strains grew. And these are 2 test plates. So isolates were deemed as having high activity or being highly inhibitory if the growth inhibition from the streak to where the culture is was greater than 5 millimeters. We also said an organism to be highly active has to be able to kill more than 1 pathogen. And we used that to identify which organisms we would carry on for the remaining of the testing. Katrin found that these isolates were very active. Nearly 80% of the isolates we isolated from Ferox had activity, and 70% of the Southerlandia isolates had activity against multiple pathogenic strains. All the isolates which showed activity using the perpendicular streak were then further tested using your standard overlay on the different media, and then we also identified them using 16S sequencing. All these findings together were then coupled to try and decide which were the organisms we would take through for full characterization. In the end, we selected 6 isolates to take through for full characterization. 4 of them were aloe isolates, so it's A3, A37, A81, and A159. And these were selected based on the fact they had very potent activities against several of the test strains. We also decided to include 2 of the Southerlandia isolates, which had very low activity and only activity against Bacillus, but based on some other tests, they looked like they were novel, and they were leaf endophytes and a seed epiphyte, which made them really interesting. So we decided to include them in our 6 champions for complete characterization. So before I discuss the bioactivity results, just a little information on the identification of the 6 top isolates. So strain A3 was isolated from the Aloe rhizospheric soil. It was formed these very pale colonies on most media, and you can see that he's bald. It produced very sparse or no aerial mycelium on most media. Based on phenotypic differences as well as the low 16S, so 16S was only 97% to its neighbor, we propose that A3 is a novel species, and we propose the name Streptomyces albertinensis for this strain. Similarly, isolate A37 had low 16S similarity. The similarity was only 95% to its neighbor. And this bug also had some really unusual chenotaxonomic features. And it points to the fact that this is definitely also a novel species. And we therefore propose the name Amycolatopsis museum in facium to refer to the popcorn-like appearance of these colonies. So this Amycolatopsis also had really potent antimicrobial activity against a whole suite of gram-positive organisms, including Mycobacterium aurum and the Mycobacterium smegmatis. You can see when it tested against M. aurum, it nearly cleared the whole petri dish for the zone of activity. Interestingly, this really high activity was only observed when the organism was cultured in ISP2. So it clearly shows that media composition has a really strong impact on the production of secondary metabolites in this strain. A37 also had really high levels of siderophore, which you can see over here compared to the control, the zone that it was produced, again, cleared the plate. As no antimicrobial activity has been reported for any of its nearest neighbors, we are really interested in what compounds this bug is producing, especially the fact that it had activity against 2 fast-growing mycobacterium. So in future studies, we are planning to extend the testing to include slow-growing pathogenic mycobacterium, including obviously Mycobacterium tuberculosis. Then the 2 maybe not so interesting of the top 6. So the sequence similarity between 2 of the aloe isolates, A81 and A159, was 99%. And these organisms also had very similar phenotypic features. When we compared it to its nearest phylogenetic neighbor, S. californicus, we found the similarity was 99%. After doing whole genome sequencing and determining the ANI, we found that the ANI between A81 and S. californicus was nearly 98%, and therefore these could not be recognized as unique species, and therefore we propose that both of these strains belong to the species Streptomyces californicus. And the last 2 of the top 6 were the Southerlandia isolates. So based on our preliminary analysis of these 2 strains, they are also likely to be novel species. So the ANI between S149 and its nearest phylogenetic neighbor was 84%, and there were several really marked phenotypic differences. And so for that reason, we propose that this is also a novel Streptomyces species, and we're proposing the name Streptomyces the Southerlandiae for this bug. And the last species is S173. This organism was found to be an Actinomadura species. It had some really distinctive phenotypic features. And then when we looked at the GRSB, when we compared the genetic distance between this strain and its nearest phylogenetic neighbor, we found that it was below the 0.02 threshold that they recommend, which would support the fact that this is also a unique species. And therefore, we're proposing that this is a novel Actinomadura. So then we moved on to the genomic screening part. So we've started by doing a PCR-based screening with your KS1 primers. The results from these sequencing revealed that 5 of the 6 strains likely harbor at least one PKS type 1 domain. The Amycolotopsis strain A37 was found to have a domain most similar to the NICE-B gene, which is involved in the synthesis of nystatin. Nystatin is a polyene macrolide, and this has really potent antifungal activity. Similarly, Streptomyces A81 had a domain which had 88% similarity to the RFA3 gene domain, which is involved with the synthesis of the antihemolytic drug Ivermectin, not the other popular Mectin drug at the moment. And as the main research focus of my group is plant growth promotion, the fact that there is a potential antifungal as well as antiparasitic activity in these bugs is really exciting. And then we are investigating this further at the moment. So we sequenced the genomes of 3 of our isolates. However, after processing, we found that there was insufficient coverage for the genome of A159. As we use the Nextera XT kit, which is a transposon-based kit, this kit can really struggle when you have high G plus C organisms or those with really complex secondary structures, which is likely for the reason that why this sequencing failed. So therefore, in the future, we're planning to repeat this sequencing using a non-PCR based method, so either a nanopore or the Nextera Flex kit. So the specs for our other two isolates, A81 and S149, were acceptable for the later analysis. So then just a little bit of results from our annotation of our genomes. So annotation of the draft genome of streptomycete A81 indicated that the strain had a approximate genome size of 7.3 megabase with a GC content of about 72%. It had 7,363 coding sequences, and these genomic features were similar to those reported for its phylogenetic neighbor, S. californicus. The assembled genome was found to have about 18% of the sequences being protein coding sequences, which could be classified into subsystems. So when you looked at the subsystem analysis, there were genes identified with secondary metabolism, and those genes were particularly those associated with ribosomal natural products. So those included 17 sequences which were involved with the lanthy peptide synthesis, as well as 10 that are involved with the synthesis of thiazole, oxazole modified microsins. So the presence of these lantharonine enzymes indicate that this strain has the potential to produce lanthy peptides, and from further analysis of the sequences, it looks as though it could potentially produce class 1, 2, and 4 lanthy peptides, as well as potentially other natural products. This is interesting because to date, the products of streptomycete class 1 lanthy peptide biosynthetic gene clusters have not yet been identified. So the genes have been identified, but the products have not been found. Investigations into the genomes of strains A81 and S149 using Antismash revealed that the isolates had the genomic potential to produce a wide range of natural products. And genes involved in the production of antibiotics, anti-tuberculosis drugs, anti-HIV compounds, and anti-tumor drugs were all identified. So when we look at just strain A81, numerous biosynthetic gene clusters were identified. So in addition to the usual PKS and NRPSs, we also identified the 2 lanthopeptides, which were also previously found using RAST. The strain was also found to have genes that are involved in the production of some plant metabolites. 2 of those metabolites is your alkyl resonal type antibiotics, as well as neuringin. This is particularly interesting as these 2 metabolites are actually produced by the host plant, Aloe Ferox. So if we look at one region that was particularly interesting in the strain, that was region 499, which showed similarities to a type 2 PKS biosynthetic pathway, and particularly those genes that are involved in the biosynthesis of grisierodin or rubromycin. Now, grisierodins are hydroxyquinone antibiotics, which were first isolated from a really red substance produced by Streptomyces californicus. Grisierodins are a family of modified aromatic polyketides, and these compounds have really great structural diversity. And this is due to enzymatic oxidation reactions, which modify the compound and also influence the bioactivity. Now, some grisierodins have been shown to inhibit HIV reverse transcriptase, as well as human telomerases. Rubromycin is a related compound, which was also isolated from a red substance produced by Streptomyces calinensis. Rubromycins are anti-tumor compounds, and they have these really complex molecular structures. It is worth noting that the solvent extracts of our bug, A81, is bright pink. So if you look at the results for your Kirby-McCullough here, you can see that the little discs here are stained pink. And these extracts did have quite strong activity against S. aureus, and anti-S. aureus activity is one of the reported activities for grisierodin. Given that this strain A81 is so closely related to the Grisier rodent type producing strain, we think that it is highly probable that strain 81 does produce one of these complex aromatic polyketide type antibiotics, and we hope to investigate this further. So then looking at the whole genome sequencing of streptomyces strain 149. So this genome of this organism was a little bit bigger, was 8.3 megabase with about 71% G plus C content. Subsystem distribution according to RAST was the same as what we found on A81. We were able to identify 4 genes involved in secondary metabolism, and all of these genes were involved in auxin biosynthesis. Auxin is a plant hormone. It's derived from the amino acid tryptophan, and it's produced by plants, and it improves plant growth and development. Now, streptomyces strain 149 had all 4 genes, TRPD, TRPA, and TRPB, that's involved in tryptophan biosynthesis and needed for auxin production. Now, studies have shown that some bacteria produce auxin, and these are able to stimulate seed growth, root formation, and root proliferation. And as streptomyces S149 is an endophyte, it would be really interesting to see if this organism is able to stimulate plant growth, especially since it produces really strong siderophores, which can also be seen from the systems analysis. So when we looked at Antismash for this organism, they predicted 39 biosynthetic gene clusters, including your terpenes, your lanthipeptides, lasopeptides, and butyrolactone. So just one of these is looking at the terpenes. So there were 5 genes clusters associated with terpene biosynthesis identified in the genome. One of the clusters had 100% similarity to the genes involved in the albaflavanone production. Albaflavanone is a ketone type antibiotic, which was isolated from streptomyces, and it has strong antibacterial activity against Bacillus subtilis. Researchers have found that this albaflavanone biosynthetic pathway is highly conserved in streptomyces species. Based on the fact that S149 does have potent antimicrobial activity against the Bacillus cereus, and the fact that this pathway does tend to be so well conserved, we feel that it is highly probable that this strain is producing an albaflavin type compound. One of the other terpene gene clusters that were identified in this organism had showed 100% similarity to the gene cluster involved in the synthesis of isorenitine, as well as 87% similarity to some other carotenoid genes. The isorenitine is a carotenoid which is a potent antioxidant, and these are generally produced by your green photosynthetic bacteria. Although the biosynthetic pathway genes cluster for this compound has been identified in several streptomyces species, as yet the actual activity has not been found, and all these pathways are presumed to be silent. Now, as Southerlandia is known to produce potent terpenoids, including your Southerlandins, these potential terpene gene pathways will be investigated in more detail to see whether any of the shared chemistry can be observed between the metabolites produced by the plant and this plant endophyte. So just in concluding remarks, so several actinobacteria with really interesting bioactivities were identified as part of the study. Genome analysis suggests that the actinobacteria do harbor the genetic potential to produce a wide range of bioactive compounds, including those with anti-tuberculosis, anti-HIV, and anti-tumor properties. And in addition, it is possible that some of these isolates are producing secondary metabolites, which could be benefiting the host plants, and including things like producing antifungals, terpenoids, or auxins. additional work will focus on characterizing some of these identified compounds. And in conclusion, the study confirms that these indigenous South African medicinal plants are definitely an untapped source of novel natural products. So I would like to thank my researchers at IMBM, my collaborators at CPT, as well as the NRF for funding, as well as Organic Aloe for supplying us with plants, and Parseval for supplying us with Southerlandia. And so, yeah, so most of the work was done here by Katrin, and this is my current research group, properly social distancing. Thank you.
Thank you a lot, Bronwyn, for this very nice and very broad-placed presentation. And of course, also thanks to your PhD student directly to actually providing this data for you to present to us in her stead. So it seems like we do not yet have questions from the chat. So I will, of course, provide one to you. First of all, I was intrigued. So you found a lot of potential gene clusters placed here that you presume to be silent, yeah, in your strains. Are you also planning to look into activating those clusters by molecular biology approaches, or?
So one of my current students, their research is actually using an OSMAC approach. Using different media to try and see if we can activate some of these gene clusters. The moment is because our honors students are only in the lab for 6 months, so we had to do something which could be done in a short amount of time. So we went rather with just the culture-based approach. That's what they're going to be doing this year, but I'm hoping next year with one of our master's students to look at more of a molecular-based approach.
All right. I mean, sure, it was already, sorry, are you finished?
No, yeah.
All right, because it was quite interesting to see actually that, I don't remember directly which one it was, but there was this huge impact of just using ISP2, I think it was, and only then you got this this huge zone of inhibition.
Yeah.
So it would be interesting to see what else you can do with actually playing around with the media, and then of course later also with genomic, with molecular approaches. So we have now received a question from the chat from Shiro Pan. He is thanking you for the nice talk and has a technical question about your CAS tests, yeah, which is how long do you incubate your plates and under which conditions do you see a clear reaction?
So we do ask for 14 days. We tend to just incubate ours at room temperature because most of the bugs prefers to grow at room temperature. So if we have to put them in an incubator, we put them at 28, but we just tend to put them at about 22 degrees for 14 days.
Right, thank you. We've got another question from Jonathan Villalobos. Asking, have the strains been tested against plant pathogens? Which is a very nice question. I also wanted to ask you actually.
So yeah, my students in the lab are busy actually growing their plates now. So we are going to be looking at the activity against Fusarium. So that's one of the big, especially it's Fusarium that affects wheat plants in South Africa. That's actually also going to be part of a proteomic project. So they're going to be looking at seeing if they can look at the plant proteome and the Fusarium proteome when they're grown in conjunction with these bugs. All
right. There's no other question yet, so I think I have a chance again, which is you showed which strains you isolated from your different from your different samples, and since you actually isolated 2 probably novel strains, I would have been interested in knowing, have you found these new strains on multiple samples? So could you guess if there is an actual connection between those strains and your plants you're looking at?
So the 2 strains which aren't novel, but they look as though they're actually the Streptomyces californicus. Those were actually isolated from two very distant aloe plants, fields. So we looked at, when we isolated from the aloe, we looked at two farms which had more like commercial planted plants. So those plants were about 10 years old. And then we also went, the farmer took us to an area where these weren't cultivated, they were just wild plants. And he estimated that some of those plants were about 50 years old. And we were able to isolate that same streptomycete from these wild, really old plants and this cultivated plant. And it was probably about a 15-minute drive between the two farms. So which was interesting that you can find the same bug, which is very close 16S, from two quite distant plants. We are looking at, for that bug, is actually designing specific primers and then actually doing a PCR-based approach, screening soil from a few of the other aloe plants we have to see if we can actually detect the signature for that bug.
Cool. We actually did also receive another late question from Rohit Kumar. He's asking, have you identified any plant growth promoter compounds from these strains?
We have done a preliminary experiment using broad beans. So it was just a very short experiment, but we tested two of the isolates. For IP reasons, I'm not allowed to say which two. And those, we just prepared a culture filtrate of the bugs, and then we soaked the seeds in the culture filtrate, the beans in the culture filtrate, and then we germinated them. And both the bugs were able to increase the time it took for the bean plants to germinate and reach what we call the V2 stage of growth. And there was also a significant improvement in the amount of lateral roots that it formed. Two of these bugs definitely have shown that they have potent plant promotion properties. That we're going to look a lot more to see what exactly about those bugs were improving growth.
That's actually really fascinating. So just a question of interest from my side again, because you did mention in the beginning that there are examples of actually soil root associated bacteria actually producing plant hormones.
Yeah.
Can you tell if so are these actually plant hormones that are also produced by the plant itself, or are those derivatives or something? And if so, can you guess where those bacteria actually got these the machinery to actually produce this? Is this some kind of lateral gene transfer or?
So there have been studies with in Arabidopsis where they knock out the auxin gene in the plant, and then they put these auxin producing actinobacteria in it, and the actinobacteria is basically able to rescue the plant, which doesn't now have naturally produced auxin. Structurally, from what we've read, is for some of those hormones there is very little difference between the bacteria that's produced the auxin, or so things like IAA, or is another plant hormone, which it's been shown the bacteria can produce, which is the same as what plants produce. One of the aloe isolates produces a very one of the terpenoids, and we've been speaking to a chemist, and he thinks that potential structure of that terpenoid looks as though it could actually be like the starting scaffold for a metabolite that's been produced by the aloe. So it's relying on the bug to produce a kind of starting block for one of the metabolites it makes. I suppose it's a bit like a chicken and egg. You don't know what came first, but yeah, we have found when we've looked at genomes of 1 or 2 of these of our other bugs isolated from, we have another project looking at Rooibos isolates, so it's another South African plant, and there are 1 or 2 genes there which look very plant-like. So I definitely think there's been a little bit of sharing is caring, sharing genes between bugs and the plant host.
That's really fascinating. So I think, oh wait, okay, so we don't have any further questions. I think my interest has been satiated as well. So I guess we will stop the discussion here. Of course, best of luck for you with your projects going forth. And I guess your PhD student already is very lucky having this nice of a project. And before we go offline, I would also like to promote our next speaker, which will be actually our host at the moment, Molly Keith Baker. She's also part of the committee organizing these e-seminars, and it will take place not next, but the Tuesday following that. I think that's the 13th of July, and you will receive additional information about it on our Twitter page and also on via the email newsletter. So keep your eyes open. Will be definitely also an interesting talk. And until then, I would say have a nice day and see you in 2 weeks.